Our next speaker is Yuriy Chaban from DLS eBIC. And I'll give it a moment to get set up.
Thank you very much for introduction. I would like to start my update with a set slide. So, in memory of Karen Davis, who was a brilliant scientist and wonderful colleague who contributed a lot to world science. And once she joined eBIC and Diamond in 2021, she was very busy to contributing to activities at Diamond and eBIC, including management and development of different routines and pipelines and mentoring people. And Pejun is going to make announcement about how we would like to remember Karen later in today at the end of the session. So now let me switch to eBIC updates. So first I would like to showcase our new solar panels on the roof of Diamond Lighthouse light source, and it produces about 2.7 gigawatt of energy per year. And I learned yesterday on Thermo Fisher seminar that Thermo Fisher estimated that an average cryos requires about 35 household worth of energy. So this provides about 700 household worth energy, and I think eBIC is quite covered, although it's only 5% of Diamond needs. So then the output, we still continue like with steady streak of publications, which currently average about 50-60 per year. And I think this year is also going to match this trend. That is mirrored by depositions. As of now, we have over 1,400 depositions in EMDB and over 800 depositions in PDB data bank, which is amazing. And thank you for everyone who was busy using eBIC and depositing those structures. Our capabilities, we started as a facility with only one working cryos and then rapidly expanded to 4 instruments. And since then, we were busy to add functionality to actually use these cryos in best way possible. So we're adding new workflows. So you not only can do single particles analysis, but many other things. We can analyze purified complexes, single particles, purified viruses with both single particles tomography. You can also use cellular tomography and microcrystals. We do not provide for typical back session ability to prepare samples like freeze it, but one can apply for chameleon use. We can have this fast freezing technique, which was showcased yesterday. And then it goes into cryos microscopes, and you can do single particles tomography and crystal analysis. It is also combined with if required, it can be combined with a cryoclam and feed milling to make lamellas. Our Oculus also has Meteor for in chamber fluorescence if you would like to validate your lamella before you go to the microscopes, which also helps to improve efficiency. We also offer, in partnership with Rosalind Franklin Institute, ability to use volume imaging on Helios Hydra. We expect that people using main eBIC facilities through academic route will publish the results. However, if you're coming from a company who would like to keep things proprietary, you can access eBIC for Industry, which has a great team and has dedicated glaciers and cryos microscope, and they offer a full range of techniques, including freezing, screening, sample assessment, collection, processing, and analysis on greater level than we would normally offer for academic user sessions. Science highlights. Single particles still provide a steady trickle of great results. And here, yesterday you heard about USP1 protein, so that's another member of this pathway, which finds merged DNA and recognizes different places on DNA. And the other work was regulation RAD51, which is also recombinase and important for health of the cell and DNA repair. The third project was, and actually it's the greatest resolution so far obtained on eBIC microscopes for like a science project. Here it was everybody's favorite beta-gal, but it was from a plant pathogen, and we were able to see, well, our users were able to see glycosaurine bound in the molecule at 1.1 angstrom resolution. So next is example from tomography, which used light microscopy and tomography to study microtubules and actin bound inside the microtubules in the thrombocyte cells. And the next example is actually using and developing a workflow for studying viral infection in nucleus. So here it's the method was developed to study to improve the study of HIV infection and the whole workflow, including correlative light microscopy, Fib milling, and tomography. So you can see that that's images in our meteor in Oculus. And using like hundreds of tomograms, it was possible to see different stages of HIV interaction with nucleus. And this work is currently being reviewed, but it's available in archives. And last but not least is work which was showcased yesterday. That's hemozoin crystal in malaria parasites, which also won the Improved Microscopy Society on Second Paper Award. So we heard a lot yesterday about pipelines, and I think today the first talk called the trend. Pipelines are important. This is why we invest a great deal of time and resources into developing new capabilities there. But it all starts with backend. So we do, our data analysis team developed a tool to look into Thermo Fisher Health Monitor and output information conveniently, so we can keep track of health of our instrument. Also, we have back-end software which is available for local contacts, and and data analysis team which allows us to track health of your data acquisition and if data analysis has started. But please notify us if you see something goes wrong, because we may not be able to capture every possible problem. The service-based tomography pipeline was developed and published last year, and we advanced this to work. So at the moment, it also includes membrane sag to segment membranes. While it doesn't work perfectly, it can still provide valuable information for your session on the fly. We're also working on the particle picking, and I will stop at it later in my talk. Single particle pipeline, it's based on Colin Palmer script, and with help of Matt Edanza, we could make it more usable into eBIC environment. And now it also follows the service-based approach. So essentially, it runs in highly parallelized way in the cloud. The output, this is like from a live session, so it produces reasonable quality 3D class averages, which can then be taken into further analysis. So how does it work in more detail? The main difference from reliant approach is that it starts from a single micrograph, and every micrograph is analyzed in a parallel way, so we don't have any latency there. It runs ice thickness estimation, although at the moment it's not linked directly in with further analysis, but still can give you like a figure which might be useful in assessing quality of your data in various spaces. Then it follows like standard routine with CTF find and particle selection. And then data are combined to determine particle size using Crayola algorithm. It's based on 80% of data from first, if I remember correctly, like 50 big micrographs. And then particles are extracted and batched into 50,000 particles. 2D line 2D classification is performed on every batch. That's also parallelized now. And then we use the line class ranker to select the best classes. So the Crayola step for machine learning based particle picking and line class ranker, we found it very important to actually give us meaningful result and remove outliers, which would otherwise make us burn time and don't produce a great result. So then particles are rebatched, so we get about 200,000 particles, and reliance for declassification is performed. It produces it's important to mention that initially we bin our particles quite harshly, Nyquist about 8 angstrom, just to get everything fast. At this stage, we still can see like general outline of particles, but we don't expect the resolution to be great. However, if it reaches 11 angstrom threshold, then particles are re-extracted with 2 times pixel size, and then 3D refinement is run, which gives a greater resolution. However, as we found, it's only roughly about a quarter of data reach this stage. So we started to search what can possibly go wrong. And if you look at 2D classes, like this example of Empire dataset, and you take like a seemingly good class, which would definitely make into class selection step. So you look inside and you realize that quite a few particles there might be just bad, and others can belong to not belong to this pose, which is probably not a problem if you have homogeneous, well-behaved data set. But if you have a heterogeneous population, that might be a contaminant or some breakdown fragment. So in order to deal with this, we developed, with help of CCP-EM and SciML group at STFC, a protocol for automated filtering of particle images. And it starts with denoising step, which uses variational autoencoders. We call it cryo-VE. You take particle images after they've been 2D classified, so it fits nicely in a pipeline. And we do need to apply poses here, because otherwise it's difficult to treat the particles. But it can give you, so it can give nice cloud. And if you then look at your particles, they are denoised. You still can see the particles, but background is lower. So then these particles are taken into a cryo-DAN approach. So it is domain adaptation network, which uses particles with denoised and reliant class ranker annotated data set, because particle annotation is a bottleneck. So because we improved sync noise ratio, we can use these particles more efficiently to make decisions. And in this way, we can discriminate particles rather than class averages on early stages of analysis. As usual, the score is an issue. So we need to accurately determine what score is correct, because it just gives you different type of score. And by looking into 2D classes and plotting membership, we assume that if half particles from good classes make it through, that's probably about the right score. And if you look at bad classes, you see that some classes which probably were wrongly assigned can actually be rescued in this way. But yeah, there are some also outliers which going through. Testing it on Empire data set, we can see that it performs on par with ClassRanker with slightly better resolution, and it helps to alleviate the preferred orientation, which is obvious when you use all particles from a 50K subset. On other data sets, which we took from Empire as just raw data, it performed to various extent, but we think that our pipeline, which uses Crayola, might already beautify our data set, so it's probably better than it might be initially. To deal with this issue, we've taken deposited data set where particles were picked by the user and deposited to Empire. And in this case, we can see that for some data set, you can get approximately the same resolution with much less particles, like 30%. With others, better data sets, you need 90% of particles to reach similar resolution. As Colin already mentioned, I have a poster in poster session 2. So if you're interested about this method, please come and talk to me. So Colin already mentioned that it is implemented in CCP-EM package, and we're also testing it in our pipeline at the moment. So what we have learned, if you run it on like 50 data sets from the user, you see that about a third of the data set have improvement. So here we needed to use not only, our first approach was to use directly CryoVAE ClassRanker, but it seems that so just CryoVAE and CryoDAN, but it looks like ClassRanker is also required because sometimes you have an outlier class which produces good quality particle but doesn't belong to the data set. So in reality, the score which is calculated by multiplying ClassRanker and CryoDAN score performs the best. But in some cases, CryoDAN alone was able to make the difference. So about third data sets, which were a border about 11 angstrom, performed better in this case. And we think that might improve possibility to push data into a refinement stage. And it also seems like for some data set, help to alleviate preferred orientation as well. But we're still exploring this aspect. Right, so another thing which our data analysis team was working on was applying symmetry. So in general, people not always tell us what symmetry we need to use. So it would be great if we can do it on the fly. Our data analysis team came up with the idea that we can apply symmetry to the models and then find the best score here. So you see that's the initial C1 symmetry refinement, and then different type of symmetries were tried. So C5 symmetry performed the best in this case. And then when we apply this during refinement, we get a better model, which hopefully helps people to analyze the data better. Vinod yesterday walked us through our new improvements in pipelines, and that in displaying result of our pipelines. This is you're probably familiar with is Pato, which web tool to show all the results from our single particles and tomography refinement, so in tomography pipelines. The new important capability is that you can now display atlas and check where you collected your data, and that is stored, because in a typical EPU and tomography session, it's gone as soon as you stop the session. It's very difficult to display back. You can also generate report and to keep track of everything. As we get more and more samples, we get faster and faster. We need to keep track of your experiments. And often you don't need to invent a bicycle. You can just look back into what you used and what you got. And especially if you have like different people working on the project, and they can just check that and find a way, because we have ability to input this information and make it available via Scope now, Scout. That will be online, will be main tool how you send us samples and put information in very near future. So if you have any questions, would like to try hands-on, Vinod and Guilherme spend a lot of time developing it, and Vinod is here and you can approach him and check it hands-on on demo version. Now, what does it give us? Instead of having separate tools for shipping sample, analyzing sample, now we have an environment which can combine all this information together, and you can annotate your samples and then track everything, and hopefully it will help people to make informed decisions. But can we take it one step further and actually help reduce load on user and help to make this decision? So Carl Morris, when he was still working at eBIC, came up with a brilliant idea to use this workflow to actually use data collection and data processing result to predict best places to collect data. So we set up to test this, and we wanted to see if we can use our on-the-fly processing result to train a neural network model. And our goal was to characterize and choose data collection targets based on our knowledge. So that's a demonstration how it performs on atlases. So you can see this atlas from different types of grids with different mesh size and hexafoil grids. And you can see that for most of them, it does very good job in predicting good areas. So here, the red circles are where ice is too thick. The blue circles where squares are too small to collect data from. It will be just waste of time. And then green, which has potentially good areas for data collection. So we still testing this approach. Here is basically what we hope to achieve here. So we can label data based on processing results, and here where Scope can actually help, because we can make it more precise for your type of grids and type of sample. And after data collection is initialized, we use pre-processing results to update our models, and then we update acquisition area based on Thermo Fisher Scientific API. Here's a caveat though, we can't do it fully automated because there's limited things which you can do with Thermo Fisher API. So it only allows us to prioritize targets. We can't completely operate the microscope and tell it what to do in intended mode. So user would still need to select some reasonable picks before this automated pipeline of smart-EM will kick in. So in summary, I would like to emphasize that we continue to develop new workflow, and please visit our website to see what's been added. And we also have access to instrument outside of eBIC. So this is, for example, we collaborate with RFI and SPT LabTech for using Chameleon. We can collaborate with RFI for using Helios Hydra. And we had also users who came and used B24 and eBIC in conjunction, so they can characterize samples using cryo-ET and also using X-ray tomography to have wider concept and context in a cell. We are working with our collaborators in investing in development of automated pipelines and interactive monitoring software, and also developing other routines which can help with data analysis. And we develop integrated workflows, and I would be nice to hear feedback about that once we're all scoped out, because there will be some period where we will be actively trying to modify things and tailor it together. And Smart EM promises to, and I think other people developing similar suites for automated data collection. So we're probably looking into a new cryo-EM revolution pretty much soon when most of things will get automated. And listening to community yesterday, there were different concerns raised. So sample preparation, we're working on it, and we know that this is a bottleneck. We're working on a commissioning waffle method, which will we still can get in touch. I think if you think you have good samples, please get in touch with James Gilchrist. But we hope that it will be available for like standard sessions in autumn this year. Automated particle picking on subtomogram averaging. We were testing Crayola general model for that, and it provided good result on single particle like specimens. It provides reasonable result for some lamellas where you can distinguish particles, but as it being a general model, it is not very specific. So it still might work for some project though. Hands-on data processing, that's also in our plans, and hopefully we'll hear more from us about that. We plan to make available soon. Acknowledgements, we thank all our collaborators for great work. We couldn't be able to achieve most of that without help of our collaborators from different institutes and our funders, which helped to work on this project. And also wonderful eBIC team and other Diamond teams which support us and help us with advice and resources. And thank you for your attention.
Thank you, Yuri. Do we have any questions? Somebody, my eyesight's terrible, but there is pointed somewhere.
Yep. Okay.
Right.
Hey, thanks for the talk. How do you do the automatic symmetry determination? Is it like a new tool, or is it just trying different symmetries and finding the best matching one?
Finding the best matching one. We're trying, yeah, that's exhaustive, but after you have a model, it doesn't take much time. So we don't do refinement, we just apply symmetry and compare the models. And then the mean square difference score is used to decide.
We also plan to do the handedness estimation, I think sometimes, because you have this 50% probability. Maybe it's better to also have this. I think there is a tool for this as well.
Maybe, but I don't. It's more interpretation. We're not there yet. So once we get consistent, then we will get high enough resolution, so we can move on to interpretation step that will become critical. Yes.
Any other questions?
Thanks. Nice talk.
Thank you.
I like the idea that you can obviously do on the fly choosing of areas, like the good quality areas and bad quality areas. Do you see a future where you can take the PATO output and then decide when to stop collecting based on achieving a certain resolution, and then you can feed that back, switch to a new grid, and then repeat the same process again?
Potentially, yes, but for this we need a consistent return of high quality models. So that's why we invested into sorting particles, so we can push like at least 50–60% of our data to refinement stage. When you can get resolution, you can say, yes, that's the model that produces good result. That's your ground truth, if you want to call it that way.
Good questions? Great. So thank you very much, Yuri. Let's thank him again.